Problem of resource allocation in neuronal computation. Our brain works constantly, even when we are asleep. Just as a computer consumes electricity, our brain consumes molecular resources. And because it thinks all the time, it also consumes all the time. So how much does thinking cost? Let's take just one brain cell, for example. In the brain, every single neuron makes numerous contacts with other neurons. As a consequence, a single neuron hosts about 10,000 synapses. There are about 100 billion neurons in the brain, giving rise to a quadrillion synapses per person. Each and every one of the synapses contains numerous copies of protein molecules. Proteins are made, used, and degraded as they become old. With an average protein half-life of 5 days, every 10 minutes, a synapse needs to renew at least one protein copy out of its 10,000 proteins. And this is the protein cost of thinking. A neuron must constantly supply its 10,000 synapses with new proteins to maintain parallel computing and to support the needs of the individual synapses during learning and memory, a process known as synaptic plasticity. Very much as the state government has to allocate resources to cities and from cities to individual households, a neuron must allocate its protein supply to its vast population of synapses. But how? Is the synapse society egalitarian or capitalistic? To answer this question, we label newly synthesized proteins with a DNA barcode, which can be spotted by its complementary sequence carrying fluorescence. Now, with the help of single molecule microscopy, we can locate each individual new protein supplied to each individual synapse. Now, we can begin to understand how a neuron manages synapses to process information, especially during learning and memory. And perhaps one day to understand how the brain creates a memory.